Hello, ladies and gentlemen. Welcome to this session where we are going to discuss today about how neurons get predisposed to assume a particular functionality in the brain and in nervous system. So let me share my screen. Yeah. So we are going to talk about predisposed neural interconnections through growth dynamics from fetal stage. Okay. So yes, this is a picture showing the complexity of interconnected neurons. And this is the eighth talk of this series. And previous two talks, we have talked about neural intelligence, how neurons work, how they can be modeled on computer with electrical circuit equivalent, and how does the human brain work, what are the various association areas, what are the various processes going on in human brain. And now we are coming to the basics of it, that what decides the input output connectivity of this intelligence control. Okay, so predisposed neural interconnections through growth dynamics. uh, and just to brush up the previous talk, we have talked about various senses, and we were worried about particularly the speed of transmission of electrical ionic current through processes like these ones. Yeah, so we talked about it, about the speed of propagation. But just to give you a realistic perspective, considering, let us say, the vision system, for example, what is the response time of a vision system, of human vision system? So to answer that question, we have to ask what is the persistence of vision. So physics tells us that if it is more than 16 frames per second in a movie, then we see it as a contiguous file, that we are not able to resolve the in-between breaks and the transition. It looks like continuous. So that's the speed of response of human vision system, which is approximately 1/16 of a second. So not too fast, of course, not comparable with the speed of electricity. It is much slower because of the post processing and because of the speeds involved and delays involved. So that's the realistic thing. And for sound, we already know that what we can hear, the ultrasonic, we can't hear, and of course, below 20 hertz also we can't hear. It's too insignificant to make out. So that's the fidelity of our sensors. Okay, now let us come to today's talk, that what are the, how does the neuron get predisposed to assume a particular role in the whole gamut of things. Yeah. So looking at these pictures, which I've borrowed from public domain, uh, okay, I'll not be dwelling too much on that. This is just to give you an idea of the complexity, right? So nerve is coming all the way from brain and passing out through the in-between gaps of vertebral column and passing like a conduit through the inner space, inner cavity of the vertebral column, stacked up vertebral column. Yeah, and it's coming out. And further, the nerves extend to right up to the fingers, you know, through the limbs, through the organs, through everywhere. Okay, so the question is, how is this information stored and realized from genetic intelligence? Okay, somewhere this, because this is too consistent to happen by chance, by fluke, or just incidentally. Yeah, so it is too repeatable for that. Yeah. So the question is, how is this data stored, how it is realized in the genetic database? Okay, so this is fairly complex. So this picture was just meant to give you the idea of complexity. Fine. So before we question how biology would have done it, let us consider how an engineer would do interfacing of such input output for an intelligent logic controller. Let us say programmable logic controller, which engineers are used to, electronics engineers, instrumentation engineers deal with those kind of controller day in day out. So how do we do it for machines? How do we do it for man-made intelligence? Okay, how do we interface various input output connections? Okay, so of course we have to have a circuit diagram showing all the connectivity of each input and output. It has to be archived somewhere. Okay. And then not just that, we have to also have a document which defines the cable routing details, that how the cables will be routed, where all they will be clamped, how they will be terminated, from where to where. Okay. So how they will be physically laid out in the architecture, that has to be defined in a document. And of course then it has to be assigned the that which input means what. Okay, what is the input output address, which address means what logic, what is the condition, does it represent in real terms. Okay, in terms of functionality. So some input output address assignment, which is input number one, which is input number two, from where it is coming, from where input number two is coming, where outputs are going as well. Yeah. So all this, there has to be an assignment chart where you define what each input output is. And then of course we have to have write a logic uh, which checks the various input conditions, prevailing output conditions, and does the logics solve the equations or statements, maybe boolean expressions or some logic equations written in some form, or statement list like a program, like a microprogram. Okay. And then those set of logics are solved repeatedly in a cycle time, and in each scan uh, we update the input output once, the whole set, and then solve the complete set of logics, and then again come back to the start, and this loop keeps on going on. That's how the logics are realized in machines. Okay. And then of course for analog controls, where we want to control, let us say, position or temperature or force or something, there is a feedback. So there is a feedback device, and then there is a commanded value, and there is a feedback is compared with the command, and then there are routines or hardware which generates an output driving signal which corrects any error between the command and the feedback. Okay. So there are so-called closed loop control algorithms where feedback based control dynamically maintains various parameters, in this case physiological parameters and also muscle movements. And it depends on the set value or assumed value of sensitivity and responsiveness, etc., that how much is that error which is to be tolerated, and what is the magnitude of action to be taken against small error, large error, and so on, and how much delayed that response should be. That is the so-called PID control, proportional integrated derivative control in engineering jargon. And the function is to servo control to maintain the commanded value stably, rapidly, without much oscillations. Okay. It should not be over damped, it should not be under damped, it should be just critically damped, so to say. So that's how an engineer would do it. But how biology does it? Let us see that. So the big question is, starting from the brain, how do the nerves know where to reach from the brain? Because as we saw in the pictures few slides back, in the brain we know there are specific association areas, that certain area of the brain controls something, it's mapped to some part of the body. Okay. And so it is known which part of the brain is statistically connected. I mean, there can be some deviation, but by far majority of the population, those are the association areas which control particular function of the body. Okay. So uh, so what brings about the interaction, the contraction and relaxation of specific muscles if command goes from certain nerve? So if one looks at the route taken by the nerves, as seen in the previous figure, it is nothing short of a wonder as to how nerves know where to go, where to connect, and how they find their route, devious route, through the spinal cord and interpret. It's not straight connection radiating from one source to everywhere, radially in a star shape. Okay, it is fairly devious route through the inner cavity of spinal cord, through the interspaces available between vertebra, and it has to reach out right up to the fingertips, following a very, very circuitous, complicated path, so to say. Okay. So, and the thing is, these roots are simply too complex and connections are too consistent. Firstly, the roots are complex, there are too many of them, and the connections are too consistent between individuals. Statistically, if you see, everybody has the nerves rooted in the same way. Now, it cannot happen by chance, and it cannot be something like it is working under influence of some field or some gradient, okay, some potential or some chemical level gradient which is seeking, because that will grow radially directly. That will directly go to from one point to another point. It is clearly a faithful execution of a well documented design. The question is where and how this blueprint is stored and how does it get implemented. Okay. So the answer to this question is it's a one line answer, which we will be explaining in some detail in the video, that the neural connections and routing happens through the growth dynamics of fetus. Okay. So it is the growth dynamics which decides these interconnections and which controls how it is going to happen. Okay. And because growth dynamics is consistent, so also these things are consistent. Now let us see what we mean. Now basically our bodies have all started from fusion of two cells, ova and sperm. The two cells quickly multiply. I'll come back to this figure, but let us start here. So human life starts with fusion of two cells. They multiply fast and form a hollow ball first. Okay, so two balls form multiple. They form a solid ball, then it becomes a hollow ball. Okay, and then the hollow ball grows a diaphragm, a septum inside, which thickens and forms three layer of embryonic sandwich. Okay, now we come here. So this is the hollow ball. Okay, this is the embryo, the embryonic membrane, which is having three layers of cells. Okay, the inner one is called as endoderm. Uh, Middle one is called mesoderm. Meso is the word for middle, the which is shown in pink, and the blue one, which is shown above, is called as ectoderm. So ecto means outside. Now this forms the outer skin. This forms the inner skin. That is lining of intestine and all the digestive tract, etc. So that is endo, the inner lining of the body. The outer is outer skin, and meso is everything in between. So all the organs by far, including heart, including blood, including muscles, including bones. That is, most of the architecture of the body comes from mesoderm. Okay, so that's how we start. But what happens is, so these are just three figures showing various stages. I'm not going into detail of what they are showing, but this is just about 17 days after conception. This is after 18 days. This is after 21 days. So things change pretty fast in this stage, on day-to-day basis. Yeah. So we were somewhere here. So the top layer is ectoderm, that is external skin or sheath. Bottom layer is endoderm, all the inner linings, and the middle layer is mesoderm, which eventually forms muscles, bones, blood, organs. And nerves or neurons actually reside on the ectoderm. That is, they are classed along with skin. They are on the top layer or of the embryo. As shown in this figure, of course, top and bottom is subjective otherwise. Yeah. So middle layer then develops a thicker layer of neurons, which is called as neural plate. Okay, so there is a spread of thicker neural plate on top of uh, uh, in between the ectoderm layer. Okay, so that neural plate is almost like a plate. Then it starts separating from the two sides. You leave the axis. Basically, in the middle, if you see, there is a process called as notochord. In some animals, that is not mammals, which do not have skeleton system, this forms the structure of the bone. This is semi-rigid rod kind of a thing. We define the axis of the body. Yeah. So notochord is still present in mammals, but of course, over and above there is a skeleton system. And once skeleton comes, then this becomes dysfunctional. This loses functionality, so to say. But yes, in the initial stage, when everything is jelly-like, this has some function at that stage. Yeah. So mesoderm forms the most of the organs. Middle layer then develops a neural plate, and then neural plate, the two sides will fold, uh, leaving out the notochord in between that is still attached. Okay, and then it forms a kind of a tubular roll. That is, the neural plate becomes like a tubular roll. Okay. Now at this stage, the neurons surfaces are in adhesive phase. That is, they will stick to anything which they are in contact with, especially certain kind of cells. So they have a tendency to catch the mesoderm cells which are lying next to them in that sandwich stage. Okay. So whatever cells are in contact with neurons which are touching there, those form an adhesive joint, a junction. Okay. So even as the neural plate folds into a roll, the attachments with mesoderm, okay, they are still retained and they form a longish process. Okay, something like axon of a neuron. Okay, so the neuron axon and dendrites grow on demand. Okay, so whatever membranes have to grow for the neurons, whatever shape changes have to happen in neuron, it happens. Basically, the algorithm is that it has to grow to neutralize the tension caused by the cell migration. Okay, so when there is a dynamics, the plate is becoming a roll. Whatever neurons were in contact with the mesodermal cells below, they still remain attached, but it grows into a small, slender process. Okay, okay, which can become later the axon, and it becomes a nerve. Okay, so the algorithm is that it remains attached to mesoderm, which becomes muscle later in its own right. Okay, so muscles and various organs, so wherever they are in contact, they will remain attached there. They will remain anchored, and neurons will migrate and they will grow so as to negate the tension, the force, and they will grow on demand. So the cells are designed to grow on demand in whichever direction it is required to grow. That is how you get a complex structure of what we showed in the picture in the slide one. Yeah. So this is the picture of dynamics. So this is the neural plate which is folding into a roll. This is a neural groove. This is a tube. Okay. This is a plate first. So this is just a neural plate. Both the ends are there. Okay. After that, these two start lifting and they start closing on, and then they form a roll. And after it forms the roll, so basically wherever the neurons are moving, they will remain attached to the layer of mesoderm, the middle layer which is forming the muscles and various organs. Okay, so those attachments will still remain clinging to those cells, and those cells also will migrate somewhere depending on the growth dynamics. So the connections will get pulled, will get drawn. Yeah, and after a stage you see some segmentation appearing here. Now this is the precursor of vertebral column. Okay, so the segmentation appears, and then later it becomes ribs and vertebra. Okay, and this upper side is the head, and this is the tail side. Okay, so this is the neural plate. This is the ectoderm, that's the outer skin. In between, you see these blue things. These are the neural plate. Thicker neural plate comes, and then it tries to become like a roll, seen from this sectional view, and then this becomes a fully closed roll, and then the growth continues after 23rd day onwards. Yeah, so neurons start getting formed as thick layer of neural plate on embryonic sandwich of endoderm, mesoderm, ectoderm, and that is the only phase where neurons differentiate and cell multiply. After that, neurons cease to multiply. Okay, so in this stage, the growth of neurons is over, and they get connected wherever adjacent cells are there in contact, and then So this is another picture which shows something similar. So this is the segmentation you are seeing. This is something like a precursor of vertebral column. This is called the tail or the caudal end. This is the cranial end or the head. Cranium is head, okay, in Latin. So this is the head side. And afterwards, all the neurons start migrating from tail to the head. They start migrating towards the head because ultimately they have to form the brain. So all the neurons have to migrate to the brain by far. So there is a movement towards the head. Okay. Now you see how the growth dynamics is. Neural plate forms and rolls up from sides and forms neural tube, and segmentation of vertebral column starts forming like cage around the neural tube. So this forms almost like a rib cage. Eventually the complete fetus also becomes tubular, becomes like a roll. Okay. All ectoderm, endoderm, mesoderm, everything forms into a tube, multi-layer tube shell, this one, roll. And the points where neurons have gotten stuck to the mesodermal cell, membranes stay attached, the anchoring points remain there, and longer processes extend from the neuron cell body to bridge the gap as the neurons migrate away. So the connections are still retained, they are still held tight, and neuron has to respond by growing. So whatever dynamics is there, neuron has to respond by growing. Okay, and it has to neutralize the force of dynamics. So beyond that, of course, 25th day, 28th day, the head starts appearing, the heart starts appearing like a bulbous process. So this is the bulge of heart. This is somewhere like umbilical cord. These are various organs start forming in between. Okay, and later the limb buds start. Head and legs start like a small bud which grows into full limb subsequently. So basically, these are all mesoderm things. So to whichever nerve they happen to be connected, that nerve has to grow as per the growth dynamics of these features, the mesodermal features. And there is a grand migration of neurons into the head. So head will swell, all the neurons will fill here eventually. Okay. So while the neural tube gets enclosed in precursor of vertebral cage, the mesoderm layer also moves by growth dynamics, forming organs, forming muscles progressively day by day. And the long processes from neurons that touch mesoderm cells become extended axons and develop later on as nerves. They get the myelin sheath and they become nerves, conducting nerves. And nerves have to pass through the gaps in between this segmentation wherever the connections are coming through. So they continue to pass through the gaps through the vertebral column. Okay. So some more picture. So basically there is a mass migration dynamics of neurons as neurons come and fill the head. Okay. And these are the segmentation and nerves are passing through and getting attached to various parts of the body, say tongue, larynx, first arch, second arch, third arch, and so on. Okay. So there is entire thing is getting transformed very fast with the dynamics where the cell neurons particularly have to migrate vigorously. So neurons have to move from tail side and they get filled into the head, to the cranium. Okay. So bunch of nerves are threaded through the inner gap of the vertebral column, the inner cavity of the lumen of vertebral column, please, and which acts the vertebral column itself acts like a flexible hose or a conduit where the nerves can suitably pass through them and remain protected from muscular movements and body postures and all. Yeah. So these are called as somites. These are the segmentation and nerves pass through them in between. Okay. I'll not go into what this picture is. I just wanted to show this part. And later as the limbs grow, the nerves has to extend right up to the fingertips. Okay. And of course, in this stage, embryonic stage, the head looks pretty grotesque with the mouth in a very ugly shape. Okay. Almost like animal. As limbs and organs grow, the attached nerves extend on demand and axons grow in length in order to neutralize the tension. And similar to axons, also the blood vessels and lymph capillaries also have to grow because they have also interconnectivity and they are spread all over. So blood vessels and lymph capillaries are also known to grow and adapt according to the growth dynamics, relative growth dynamics of tissues. But here the single cell is extending its processes as dendrites and axon. There, of course, the blood capillaries are made of multiple cells and cell multiplication takes place. So there, it is cell division on demand and capillaries develop, vessels, blood vessels develop, and they get rooted suitably by the growth dynamics. Okay. Coming to the brain starts from a tube, and then you see the forebrain, midbrain, hindbrain represented as some three lobes of the tubes, and then this is some more detail. This is some more detail. So just for, and then is the bony segmentation cage starts appearing around the cell, marking the beginning of rather restrictive vertebral column cage around. There is a massive migration of neurons towards the head, and eventually most neurons migrate into brain with lot of turbulence and churning. As the neurons flow to in the head, it is a big turbulent flow, and there is lot of crisscross. Okay, for example, uh, in the brain, the lower part of the body is controlled by neurons which are at the top of the brain, and the eyes and smell sense are attached. They are top at the top of the body. Eyes are fairly at the top near the head, but they are controlled by the bottom part of the brain. So there is a crisscrossing, obviously. The lower part of the body gets mapped to the upper association areas in the brain. And similarly, the left side of the body is controlled by the right side of the brain, and right side of the body is controlled by left side of the brain. Okay. So there is nature has made a deliberate crisscrossing when the neurons migrate, so that churning is there, so that fleeting opportunity is there to make connections with the one-time neighbor, which cross and happen to rub during the dynamics of growth. Okay. So the growth dynamics of neuron present fleeting opportunity to the neurons to make adhesive connection with their one-time neighbor, muscles or other neurons. If it is neuron to neuron, it is dendrite and axon connection. And if it is to the muscle, of course, it is nerve. So it is axon. Okay. But all these processes have to extend on demand depending on the dynamics. Okay. So since neurons have flexible shape, the connecting processes stretch and extend their slender axons and dendrites. So finally, the brain looks something like this. This is a fully grown brain, where after a lot of crisscross migration of neurons. Okay. So, and this is the bottom view of thing. And there is evidence that there is a lot of inversion of connections. That is top to bottom, bottom to top, left to right, right to left. So all those crisscross connections happen. And there are evidences, for example, as I said, left side of the body is connected to right side of the brain and vice versa. Lower side of the body is controlled with upper side of brain and vice versa. Yeah. Here we can see the optic charm, that is something connected with the eyes, and olfactory bulb, that is smell. It's right at the bottom. Now these sensors are at the top of the body fairly, it is in the head, but this is at the bottom. And something which is on the top is something which is going to limbs. Okay. Legs and lower part of the body is controlled by the top of the brain. And another evidence is that if you see the optics, anything which we see, its image is falling inverted on the retina, but we see it upright, which means that the pixels in the retina, their connections get inverted in the brain through neural connections. Again, there is a crisscross. Okay. Left side of retina goes one way, but top and bottom has definitely got inverted. Okay. Left and right may not get inverted, but top and bottom of retina have definitely got inverted. Yeah. So there is, in a sense, lot of crisscrossing during the migration of growth, the migration dynamics of neurons during the growth phase. So inversion gives opportunity for widespread interconnections between neurons. So any neuron which crosses and happens to touch during the dynamics phase, during the turbulent phase, it will establish a connection. So interneural connectivity is governed by the growth dynamics. And there are some freak phenomena because of this. For example, things which are unconnected functionally or physiologically or architecture wise. Now, what has the earlobes got to do with these organs which are mentioned here? Nothing. These are the soles, and some organs are named here. Now, this part of the foot is attached to trapezius. Now, what is the relation? There is no relation. Apparently, there is no relation. What is the relation? Okay. Now, the relation is there through neural interconnectivity. That is, some neurons which have landed here finally. Okay. Some neurons which have landed here finally, they have some connectivity to these organs, to the neurons which are connected to these organs. Okay. So, in a way, the sole, the earlobe, and the palms represent the whole map of the body, the whole map of neural connectivity, interconnectivity. Okay. So, also in hand, different zone of hands represent, they are neurally connected to different organs of the body. So, if you prick a needle here, that organ gets affected. That's how the acupuncture works. That is how acupressure works, of course. Acupressure will alter by working the blood flow around that organ, and acupuncture will directly give a nerve stimuli. Okay. So, basically, growth dynamics also results in mapped area of neural interconnectivity with inner body organ systems which have no direct functional, morphological, and physiological connection with the mapped areas. So, these are kind of mapped association areas. Okay, from neural connectivity point of view. So, something if you do here, that organ gets affected and vice versa. Okay, due to neural interconnectivity, due to neural intelligence. Okay, so palm, soles, and earlobes have neural connected intelligent linkages with the entire body. So you can treat entire body by just massaging or pricking the palm, soles, and earlobes. Okay, so therapies like acupuncture, acupressure, or even massaging has indirect influence through neural intelligence, and it alters the blood flow by indirect constriction or dilation of blood vessels in that area. As a deferred reaction from stimulus, suppose you stimulate here, as a deferred reaction, that organ gets affected through the neural interconnections. Yeah. So this is interesting, but lesser explored area so far. So answer to the question is, how do the neurons find where to go and get connected? Most certainly, there is a design blueprint embedded somewhere in the genetic code. But the genetic code is meant to operate progressively and incrementally over the then existing structures. Okay. So it's not that it's like a design engineer's blueprint where the complete finished building is shown on a drawing. It's not like that. Okay. It's not a documented design for the final product. It is more like a multi-stage sequential recipe. It's like treasure hunt. Go there, find the next instruction, do that, then go somewhere else and find the next instruction, like that. So it is a sequential recipe with defined starting and end condition, that for each stage what is the starting condition and what is the ending condition. From there to there, that recipe has to be followed. Okay. So The code operates normally at the genetic code, of course, is meant to operate at intracellular level within each cell, while the growth happens at tissue interaction level. So the relative growth of, let us say, muscle and bones will determine how they will look like, how they will get shaped. Okay. So organs, the entire growth of organs depends on the relative stiffness of the layers of cells and how they fold and how there are fibers passing through them. So, and what is the rate of growth of different layers? Accordingly, any shape can be generated. So it is additive manufacturing to the best. So as a result, the relative growth rate and stiffness of adjacent tissues, that is what is controlling the growth dynamics. That's what is controlling the force experienced by individual cells from all around, and the force which is causing the migration. So that inherently brings in some kind of mistake proofing and robustness, because anything actuating wrongly at a wrong time, out of turn, it will not be supported by the then existing conditions and the structures around. So it is simple. If you follow the recipe stage wise, satisfying all the conditions of stage transitions, then only it works. So that's what is the secret of success. That's why it is so repeatable statistically. Now, it's an interesting subject, but what is the scope for further research? Yes, there is a lot of scope. For example, suppose on a neural plate, we define the neurons or some zones of cluster of neurons, for example, which stay together. Okay. So you can define something like finite element. Okay. You define some zones on the neural plate in the embryonic sandwich stage. And you also define the adjacent mesoderm cells which are there, endoderm and ectoderm elements which are forming the fetal embryo. Okay. So the embryonic sandwich we model mathematically of which cell is residing where, which is neighbor to which cell. Okay. Then make a day-wise fetal growth model as an animation file. Okay. As a movie file representing that the growth of skeleton, muscles, and organs which happen day to day, day wise. Okay. So you make a video file. Okay. After study, you can make, of course, I'm sure it's possible to do it. And then model the connection of neurons to the neighboring cells, and as the neighboring cell and neurons themselves move, how the shape of axon or nerve will look like. Okay, so one can model that as well, and then have the modeling of kinematic pathways of how the neuron migrates through the growth dynamics and how it mass migrates into the head and how it reaches the final destination in brain with crisscross connection and turbulence, and which all neurons will cross as neighbors during that transition. So where all the connections could be established from synapse to axon. Okay. So how the IOs will get interconnected for each neuron could be modeled. So the neurons which brush during migration turbulence are identified as synaptic connections. And then, of course, one can check with the physical, biological specimens, compare and refine the mathematical model. It can be validated and verified statistically with biological observations. And then the input output connectivity thus arrived for various neurons could be used as input condition for simulation of neural learning further, so that we know which cluster of neurons will assume what role. Okay. uh, Refer to the previous talk. Which ones will have the object level memory, which neurons will have the pre-primary learning, which will have the primary learning, which will have the, you know, the higher order learning, and which will have the intellect. Okay, so everything can be modeled on computer. It's an interesting area, lesser explored yet, so there is a lot of room. With this, we end today's talk. I hope you have found it interesting. So In the next talk, of course, will be these three talks, this one and the two previous talks. They cover basically by far all the aspects of neural intelligence and how it could be modeled on computer. Okay. In the next talk, we'll deal with some other interesting topic of how neural intelligence can be used for therapies. We touched a little bit on acupuncture, acupressure, etc. But we talk about some more therapies where the medicines work through this, where the medicines work through neural intelligence rather than at hardware level treatment or chemical level. Okay. How does it work as a software debugging tool? So stay with us for the next talks. Until then, bye bye. Good luck. See you.